Hi, welcome to class. Today we're going to be covering Chapter 4 of Psych 323, that is Sensation and Perception, and this is the visual system, specifically the brain. So let's kick it off by talking about visual pathways and function beyond the retina, because the previous chapter talked about the retina, and we're moving on from that. Light is transduced by the retinae of the eyes and converted into a neural signal. The retinae begin processing the visual signal, looking for edges and coding for color. This process intensifies when the optic nerve leaves the eyes and heads for their first synapses in the brain. So, we follow the neural signal as it leaves the eye and travels to the lateral geniculate nucleus, or LGN, of the thalamus. And from there, to the visual cortex in the occipital lobe. We also consider other pathways of visual information after it leaves the eye. Ooh, and a little fun fact here is that approximately 1 million retinal ganglion cells form the optic nerve of each eye. Wow. The optic nerve and chiasm. Alright, the optic nerves leave the eyes and travel towards the brain. At the optic chiasm, each optic nerve splits apart. The optic chiasm is the location in the optic tract where the optic nerve from each eye splits in half, with nasal retinae crossing over and temporal retinae staying on the same side of the optic tract. The optic nerve of the left eye and the optic nerve of the right eye meet in the optic chiasm. Optic tract is just the optic nerve starting at the optic chiasm and continuing into the brain. The right optic tract receives information from the left visual world, and the left optic tract receives information from the right visual world. Information from the left visual world travels first towards the right side of the brain, whereas information from the right visual world first travels towards the left side of the brain. However, information from each eye does go to both sides of the brain. For example, the left temporal retina receives information from the right visual world, which stays on the same side of the brain and goes to the left hemisphere, which is ipsilateral organization. The left nasal retina receives information from the left visual world, which crosses over and goes to the right hemisphere. This is contralateral organization. And to kind of get into this a little bit more, ipsilateral organization is just same side organization. So in the visual system, the temporal retina projects to the same side of the brain. And then with contralateral organization, this is just opposite side organization. So in the visual system, the nasal retina projects to the opposite side of the brain. Now, contralateral representation of visual space is just the arrangement whereby the left visual world goes to the right side of the brain, and the right visual world goes to the left side of the brain. Oh, and a quick note here is that the brain is initially respecting the outside visual world with its representation in the brain. Alright, axons from the ganglion cells in the right half of the right retina and the right half of the left retina combine. These form the optic tract on the right, so the right optic tract. This then proceeds to the right hemisphere of the brain. Okay, now axons from the ganglion cells in the left half of the right retina and the left half of the left retina combine, forming the left optic tract. This then proceeds to the left hemisphere of the brain. So TLDR, for one, information from each eye goes to both hemispheres, and for two, each hemisphere of the brain receives input from the contralateral visual field. And this already has become a lot to handle, so I recommend this video here. It is from the channel Neuroscientifically Challenged, alright? It's just a 2-minute video. I recommend you play it from 31 second to 1 minute 11 second. It seriously does a lot of good. The basics of the video I can sum up here. I recommend you go watch it for yourself. The optic nerve leaves the eye and extends to a structure known as the optic chiasm, where it meets the optic nerve from the other eye. At the optic chiasm, the optic nerve fibers carrying information from the sides of the retina closest to the nose cross over to the other side of the brain, while those carrying information from the sides of the retina closest to the temples remain on the side of the brain where they are. This is the nasal being contralateral and the temple being ipsilateral. After leaving the optic chiasm, the nerve fibers are referred to as the optic tract. Most of the fibers in the optic tract end in the LGN of the thalamus, and from there the information will be passed onto the visual cortex. 4. Leaving the chiasm. Once the optic tract has exited the chiasm, 90% of the axons make their way to the LGN of the thalamus. Here, the organization continues to respect the left field, right field distinction. This pathway then leads to the visual cortex. However, 10% of the axons from the optic tract go to other locations in the brain. Alright, some axons go to the superior colliculus. The superior colliculus is a midbrain structure that sits below the LGN. Alright, it is an important structure in eye movements, and yes, we will talk about this later. Some other axons head to the frontal eye field region. This is an area in the frontal lobe also instrumental in eye movements. And some fibers also go to the pineal gland, and this just regulates our circadian rhythms. And this is why exposure to bright lights can help reset our internal clocks. But we're moving on. The lateral geniculate nucleus. This is a bilateral structure in the thalamus that relays information from the optic nerve to the visual cortex. The thalamus then is a large structure that serves as a relay station for a number of sensory systems, including both vision and hearing. Now, the LGN is the critical locus in the thalamus for vision. The LGN has complex functions like visual processing, but it is still thought of as a relay center because it holds the middle position between the retinae of the eyes and the visual cortices. Now, anatomically, the LGN is divided into 6 layers. Layers 1 and 2 are the magnocellular layers because the cells in these layers are large. Magnocellular layers are just layers of the lateral geniculate nucleus with large cells that receive input from M ganglion cells, which are parasol retinal ganglion cells. Layers 3 through 6 are the parvocellular layers because the cells are somewhat smaller. Parvocellular layers are just layers of the lateral geniculate nucleus with small cells that receive input from P ganglion cells, which are midget retinal ganglion cells. Kinocellular layers, then, are just thin layers between each of the 2 magnocellular levels and the 4 parvocellular layers that consist of very small cells. Okay, there are 6 kinocellular layers. 1 under each of the 2 magnocellular layers, and under each of the 4 parvocellular layers. And a quick note here is that kinocellular layers may play a role in color vision and in the analysis of contrast, possibly also motion perception. So, TLDR, the LGN is a 6-layered structure. Layers 1 and 2 are called magnocellular layers because the cells in these layers are large. Layers 3 through 6 are called parvocellular layers because the cells here are small. 6. Important LGN facts. For one, we have 2 LGNs, alright, 1 in each hemisphere. The left LGN receives input primarily from the right visual world, and the right LGN receives input primarily from the left visual world. 2. Each eye projects inputs to both the left and the right LGNs when half of the retinal axons cross over in the optic chiasm. 3. Each LGN layer receives input from only one eye. Okay, magnocellular layer 1 and parvocellular layers 4 and 6 receive input from the contralateral eye, which is just the eye on the opposite side of the head. This is also true for the kinocellular layers underneath each of these layers. Magnocellular layer 2 and parvocellular layers 3 and 5 receive input from the ipsilateral eye, which is the eye on the same side of the head. This is also true for the kinocellular layers underneath each of these layers. So, the LGN preserves information about where information in the visual world is coming from and which eye is detecting that information. Now, knowing which eye an image comes from is important in constructing a 3-dimensional image. Fourth of all, magnocellular layers of the LGN receive input from parasol retinal ganglion cells. These are M cells. Parvocellular layers of the LGN receive input from the midget retinal ganglion cells. These are called P cells. Kinocellular layers of the LGN receive input from bistratified retinal ganglion cells. These are called K cells. 7. The neurons in each layer of the LGN show retinotopic organization, meaning that retinal ganglion cells from adjacent regions of the retina connect to cells in adjacent areas of the LGN. So, the LGN has an organization oriented to the visual world. Adjacent areas in the visual world are picked up by adjacent areas in the retina, which are in turn projected to adjacent areas of the LGN. Ooh, and a fun fact here. Once we're in the brain, the representation of the visual world is more important, really, than which eye this information came from, except when considering depth perception. Moving on to the subtypes. Alright, subtypes of retinal ganglion cells. We have parcel retinal ganglion cells. These are the M cells. Alright, retinal ganglion cells that project to the magnocellular layer of the LGN. These M cells represent 10% of the ganglion cells that have high sensitivity to light. So, we're talking good light sensitivity from high convergence, which is just projecting to the magno layer. M cells receive input from many photoreceptors, including both rods and cones. So, they have large receptive fields and they are sensitive to light, but not color. M cells have lower acuity relative to both K cells and P cells. So, for this reason, the M system is associated with visual functions like light detection and motion detection. Midget retinal ganglion cells, or P cells, are retinal ganglion cells that project to the parvocellular layer of the LGN. Alright, these P cells represent 80% of ganglion cells. They possess low sensitivity to light, and they are sensitive to wavelength, which is the basis for color perception. So, 80% of ganglion cells are these P cells. We're talking about good acuity and good color vision. This is all projecting to the parvo layer. Now, midget retinal ganglion cells usually receive input from a single cone in the fovea of the retina. Carrying detailed information is necessary, in this case, for visual acuity. When stimulated, these cells show a sustained response, meaning that they continue firing throughout the time period when a stimulus is present. Last but not least, we have the bistratified retinal ganglion cells. Right? These are the K cells. We're talking about retinal ganglion cells that project to the kinocellular layer of the LGN. These K cells represent 10% of ganglion cells. They possess low sensitivity to light, and they are sensitive to wavelength, so they show some role in color vision. K cells may be more broad in their function, like color and motion, but less is known about them compared to the other cells. And again, these K cells, these bistratified retinal ganglion cells, they project to the kinocellular layer. K cells receive input from cones, but there is more convergence among these cells, so that they show lower acuity than P cells. Ooh, fun fact here. The midget ganglion cells are receiving inputs from the cones in the retina. Now, a quick note here is that the retinal ganglion cells emerge from specific locations along the retina. Those close to the fovea receive input from few, or even just one receptor cell. And those at the periphery may be collecting information from many more receptor cells. To this point, there are 3 unique pathways that get started in the retinae and continue to the visual cortex. We've mentioned these in passing already, and now we have the definitions. Magnocellular pathway, or M pathway. This is a pathway that starts with the parcel retinal ganglion cells and projects to the magnocellular layers of the LGN. And then we have the parvocellular pathway. All right, this is the P pathway. This is a pathway characterized by midget retinal ganglion cells. These are the small ones. And then we have kinocellular pathway, all right, the K pathway. It's the pathway that starts with bistratified retinal ganglion cells and projects to the kinocellular layers of the LGN. So, TLDR, the magnocellular layers are sensitive to motion, light detection, and sudden changes in the visual image. The parvocellular layers specialize in foveal functions like color, acuity, texture, and depth. The kinocellular layers also seem to specialize in color, but again, we don't know as much about these as we do about the other ones. Right. 8. Organization in the LGN. Organization within the LGN is complex but systematic, so pathways of information are preserved. The LGN maintains a retinotopic map of the left or right visual world in each of its layers. Each of these layers emphasizes different aspects of visual processing. Now, single-cell studies found adjacent cells in the LGN that respond to visual stimuli that excite adjacent cells in the retina. Important. Receptive fields of LGN cells are more like retinoganglion cells than they are like receptors. So, LGN neurons have receptive fields that are similar in characteristics to the retinoganglion cells with center-surround organization. Some LGN cells respond maximally to stimuli that are present in the center of the cell's receptive field but absent outside of the center. So, LGN neurons show specific responses to edges, spots, and gratings, just like retinoganglion cells. There are many feedback loops from the cortex back to the LGN, and many connections from the LGN to other areas of the brain. The LGN receives input from the cortex, from the brainstem, from other loci in the thalamus, and from within the LGN itself. There are more connections from the cortex back to the LGN than there are from the LGN to the cortex. Ooh, a little fun fact here is that most of what we know about the different functions of different layers of the LGN come from single-cell recording experiments using non-human animals, like monkeys. I'll talk about those later. Another fun fact is that during sleep, the thalamus is inhibited. So, unless very bright lights are shining directly into the eye, a person's eyes can be open during sleep, but they will not see because information does not leave the LGN. Information is registered on the retina and is sent to the LGN where it stops. Inhibition prevents the information from being sent to the visual areas of the cortex, preventing conscious vision during sleep. And remember, the LGN is part of the thalamus. Alright, now we talk about the superior colliculus. We have two superior colliculi, one on each side of the brain. The superior colliculus is at the top of the brainstem, anatomically just beneath the thalamus. The superior colliculus is a structure located at the top of the brainstem, just beneath the thalamus, whose main function in mammals is the control of eye movements. So remember that. The main function of the superior colliculus is the control of eye movements. Like the LGN, the left superior colliculus receives input from the right visual world, and the right superior colliculus receives input from the left visual world. The superior colliculus is part of a largely unconscious system that helps us direct our eyes towards new or approaching objects. About 10% of retinoganglion cells go to the superior colliculus. And see, I mentioned that earlier. See, it's all coming full circle. These retinoganglion cells are bistratified retinoganglion cells, part of the K pathway. The superior colliculus receives feedback from the visual cortex, and the superior colliculus projects to the kinocellular levels of the LGN. The superior colliculus also projects to areas of the visual cortex beyond the primary visual cortex. There is a retinotopic map of the visual world in cells inside the superior colliculus. Now, important to note here is that the main pathway is directly from the retinoganglion cells to the superior colliculus, allowing the superior colliculus to produce quick eye movements. Ooh, a little fun fact here is that the superior colliculus is often implicated in blind sight. We will talk about that later. Right now, we're talking about eye movements. Okay, eye movements are the main function of the superior colliculus. In general, human eyes can make two kinds of voluntary eye movements: smooth pursuit eye movements and saccades. The superior colliculus is vital in making both of these. Smooth pursuit eye movements are just voluntary tracking eye movements. So think about watching a bird fly across the sky. Alright, your eyes slowly and smoothly move as the bird moves across your field of vision. These smooth pursuit eye movements can only be made when we are watching a moving object. And then we have saccades. Okay, saccades are the most common and rapid of the eye movements. Sudden eye movements that are used to look from one object to another. We make saccades when we move our eyes from one stationary object to another. We also make saccades as we read, moving our fixation from one word to the next, just like I'm doing right now. If an object abruptly appears at our periphery, activity in the area of the superior colliculus responsible for that area in visual space becomes active, leading to a saccade. The superior colliculus also receives input from other sensory systems, especially the auditory system and the somatosensory system. All right, this allows our eyes to be directed quickly to the location of sound or touch. Like when someone taps your shoulder, you quickly move your eyes in that direction. Or if you hear a loud sound off to your side, you will turn quickly to see what caused the noise. Ooh, and a little fun fact here is that these responses in the superior colliculus are additive. So, if we visually detect something at the periphery and we hear a loud sound at the same time, our superior collicular response will be larger than for either of these stimuli alone. So, TLDR. The superior colliculus may enable some forms of unconscious vision. The superior colliculus receives about 10% of retinal ganglion cells, and the superior colliculus enables smooth pursuit eye movements and saccades. And a quick note here is that another speculation is that the K cells in the LGN project to the motion perception region in the cortex, which is area MT. We'll talk about that later. On to number 11, the primary visual cortex, V1. After leaving the LGN, the next synapse in the visual pathway is in the primary visual cortex, in the occipital lobe of the brain. The primary visual cortex is referred to by lots of names. Alright, you may hear things like primary visual cortex, V1, striate cortex, area 17, BA 17. I'm just going to use V1 and primary visual cortex. That's it. Primary visual cortex is the area of the cerebral cortex that receives input from the lateral geniculate nucleus located in the occipital lobe and is responsible for early visual processing. You can actually easily locate your own V1. Just run your hand along the back of your head, just above the neck, and at the very back of your head is a slight bump. This is known as the inion, right, in the occipital bone. Press your finger against that bone. Underneath the inion is V1. Now, because your inion is right at the center of your head, your finger will be above both the left and right V1s. Now, the occipital lobe is the visual area of the brain. This figure, right, this one right here, shows the location of V1 within the occipital lobe. V1 is the first area in the cortex that receives input from the retinae. The feedback loop is a fairly recent discovery, right, and such loops may be involved in conscious awareness. The feedback loop is just the thing between V1 and the thalamus. Thought I'd add that in there. Let's talk about mapping the eye on the brain. Mapping the eye on the brain. The visual cortex is highly organized, alright? Its anatomical structure can be correlated directly with its function. So, if we know where a neuron is, we know what it does for the visual system. V1 contains a retinotopic map with cortical magnification. A retinotopic map is just a point-by-point relation between the retina and V1. The primary visual cortex is a bilateral structure, meaning that there is one in the left hemisphere that receives input from the right visual world, and there is one in the right hemisphere that receives input from the left visual world. The left and right V1s meet in the middle of the brain. The area where they meet in the center is the area responsible for representing the fovea, or the area in space that we're looking at. So basically, the left V1 receives input from the left LGN, and the right V1 receives input from the right LGN. This means that each V1 will receive input from the opposite half of the visual world. The left V1 receives input from the left half of each retina, which corresponds to the right half of the world. Adjacent locations in the retina project to adjacent points on V1. Ooh, and a quick note here is that the V1 map is not a precisely exact map of the retina because some regions of the retina get to take up more space of V1 than do others. This is just cortical magnification. Cortical magnification is the allocation of more space in the cortex to some sensory receptors than to others. The fovea has a larger cortical area than the periphery. So, larger area for the fovea, a smaller area for the periphery. That's cortical magnification. The fovea actually takes up a huge proportion of V1 and the rest of the visual cortex. The fovea is just less than 1% of the retina in terms of size, but its representation takes up over 50% of V1 because the fovea is specialized for color vision and visual acuity. That's pretty important stuff. V1 is a 6-layered structure, like all of the cerebral cortex. So each layer is numbered 1 through 6. In V1, layer 4 is most often divided into at least 3 separate sublevels, and one of these sublevels is typically also divided into still smaller sublevels. So V1 has got a lot of layers going on. Layer 4 is the critical layer that receives input from the LGN. Alright, the 3 distinct pathways come from the retina through the LGN and then to V1. And these pathways are the P pathway, the K pathway, and the M pathway. 2 of these arrive at V1 in layer 4. These are the magnocellular synapses in sublayer 4CA and the parvocellular layer synapses in sublayer 4CB. As if it's not confusing enough. 13. Receptive fields of V1 cells. Cells seem to line up in columns that are sensitive to the orientation of objects in particular areas in the visual field. There are 2 distinct kinds of cells in V1. These are simple cells and complex cells. Let's talk about simple cells. Simple cells are V1 neurons that respond to stimuli with particular orientations to objects within their receptive field. The preferred orientation of a simple cell is a stimulus orientation that produces the strongest response. Simple cells are found in layer 4 of V1 and receive input primarily from layer 4C of V1. Simple cells have clear excitatory and inhibitory regions, similar to cells in the LGN. But, unlike LGN cells, simple cells have orientation selectivity instead of center-surround visual fields. Elongated stimuli that look like bars seem to be particularly effective stimuli for these simple cells. Some cells want dark bars on a light background, and others respond better to white bars on a dark background. This just shows a sort of color preference in these cells. The bars may also occur at varying angles of orientation. Right? These vary in a predictable pattern. This selective firing rate to the orientation shows the selectivity of the cell to orientation. Basically, the preferred orientation of a simple cell is the stimulus orientation that produces the strongest response from the simple cell. The preference is the orientation that produces a response that is strongest. Simple cells respond best to a stimulus with a particular orientation, and as the orientation gets larger or smaller, the response to the cell decreases. If the orientation is greatly off, then the cell will not respond at all. So, TLDR, V1 indicates the orientation of lines in the visual world with simple cells by having select cells respond to different angles of orientation. Wow. Talking about complex cells. Alright, complex cells are also neurons in V1 that respond optimally to stimuli with particular orientations. But they respond to a variety of stimuli across different locations. So, for example, a complex cell will respond to a dark bar on a light background and a light bar on a dark background, whereas a simple cell would respond to one but not the other. Also, complex cells do not have peak location sensitivity. Again, unlike simple cells. Complex cells will respond equally well to an optimal orientation regardless of where. Now, the important thing to note is that complex cells are found in layers 2, 3, 5, and 6 of V1, but not layer 4. TLDR, complex cells are neurons in V1 that respond optimally to stimuli with particular orientations. But unlike simple cells, they respond to a variety of stimuli across different locations, particularly to moving stimuli. Some complex cells receive input from both eyes and may be involved in depth perception. For moving stimuli, some complex cells are responsive to movement in one direction, whereas other complex cells are responsible for movement in another direction. Ooh, and a little fun fact here is that neurons in V1 are tuned to many different features, including color, motion, depth, direction, length, and size. And this is particularly true of the complex cells. Ooh, and a quick note here is that there are also neurons in V1 called end-stopped neurons. Alright, end-stopped neurons respond to stimuli that end within the cell's receptive field. If the pattern continues beyond the receptive field, these cells do not respond as greatly. Because of this pattern, end-stopped cells are considered to be involved in the detection of corners and the boundaries of shapes. And now I just have a few pointers from the PowerPoint that I wanted to throw in here because they seemed important and I didn't really know where else to put them, so here we are. For one, a bar detector in V1 specializes for the orientation of a stimulus. For two, simple cells are found in V1 and they respond best to specific degrees of line or bar tilt. And to this point, an orienting tuning curve is a graph that demonstrates the typical response of a simple cell to stimuli of different orientations. And for three, last but not least, complex cells are also often motion selective, and often more broadly spatially tuned than simple cells. So, simple cells are in layer 4, complex cells are in layers 2, 3, 5, and 6. Note, alright, this is a different area of layering than what was covered in the LGN. So when we talk about the layers that we find these simple and complex cells in, these are different layering than what we covered for the LGN. Let's talk about the organization of V1. Alright, some cells prefer to respond to inputs from one eye, and other cells prefer to respond to inputs from the other eye. This is called the ocular dominance of the cell. So, cells are organized into columns, and some cells respond more to stimuli from the right eye, and some to stimuli from the left eye. All of the cells in a given column respond to a bar in the same location on the retina, demonstrating the same receptive field, which is consistent with a retinotopic map. Now, simple cells are select for bars at particular locations. So, if you insert an electrode at a different angle, you would find cells that all respond to the same location, but for different orientations. At a different angle, the electrode might find cells that responded to the same orientation along an adjacent column. Now, adjacent columns respond to lines that are tilted only slightly differently from one another. The columns form an organized pattern according to their orientation. Ooh, and a little fun fact here is that orientational selectivity goes in one direction in V1, whereas the input from the two eyes goes in the other direction in V1. So, TLDR, we have both ocular dominance columns and orientation columns. Ocular dominance column is a column within V1 that is made up of neurons with similar responses to the orientation of a shape presented to those neurons. The ocular dominance columns are made up of neurons that respond only to one eye. These columns are perpendicular to orientation columns, which selectively respond to small variations in orientation. Ocular dominance columns alternate systematically between left eye and right eye dominance. And then with the orientation column, this is just a column in V1 that is made up of neurons with similar responses to the orientation of a shape presented to those neurons. Orientation columns change systematically across orientations. Now, a quick note here is that when ocular dominance columns and orientation columns are combined, they form something that we call a hypercolumn. Now, a hypercolumn is a 1 millimeter block of V1 containing both the ocular dominance and orientation columns for a particular region in visual space. Oh, but we're not done. We still gotta talk about the blobs. We have blobs and interblobs interspersed within V1. Blobs are areas within V1 sensitive to color, whereas interblobs are areas that are sensitive to the orientation of an object. Remember, in the LGN, there are two types of cells. We have the magnocellular and the parvocellular. These two layers divide into three types of cells in the cortex. These include the blobs, the interblobs, and the cells in layer 4B. Now, the interblob cells respond like the simple cells. They have orientation selectivity. The blobs show color responses, and the layer B4 cells respond well to moving stimuli and to stimuli of very low contrast. Taking this all together, we have a few points. For one, organization is seen in the hypercolumn. The the place where dominance and orientation columns combine. For 2, the orientational selectivity goes in one direction, while the ocular dominance goes at right angles. For 3, in each set of orientation columns for each eye, there are 2 blobs. Layer 4 runs throughout all of the cortex and cuts across all of the columns. For 4, this hypercolumn is one functional unit, processing all of the information from one region of the cortex. Now, adjacent hypercolumns process information from adjacent regions of the cortex, and it is these hypercolumns that make up the topographical map that we discussed earlier. And finally, 5, last but not least, the proportions of the hypercolumns that are active at any time will indicate the features of what is stimulating that region of the retina. Right, just a few more points yet to cover, and then we are done. We're talking about V2 and beyond. After information leaves V1, it travels to other areas in the occipital cortex. These areas are collectively called the extrastriate cortex, or secondary visual cortex. The extrastriate cortex is the collective term for visual areas in the occipital lobe other than V1. V2 is the second area in the visual cortex that receives input. V2 is often considered the area that starts with visual associations rather than processing the input. It is sometimes called the prestriate cortex. There are 3 distinct regions within V2, which match up directly with the 3 different types of cells in V1. The blobs connect to the thin stripes. The layer 4 connects to the thick stripes. The interblobs then connect to the interstripes. Now, thin stripes will have color responses, thick stripes will be sensitive to motion, and interstripes will be sensitive to shape and position. And a quick note here is that V2 cells combine input from both eyes. Other regions, like V3, V4, and V5, receive input from only some of the cell types in V1 and V2. And backtracking really quick, V5 is also known as a motion detection area, MT. I told you that would come up again, MT, long time coming, there it is. It's like 17 pages later. V1 and V2 send information in parallel to these regions. Now, importantly, it seems like V2 is involved in representing what is out there instead of actually making sense of it. It's a quick way to remember this. V2 is involved in representing what's out in the world instead of actually making sense of it. Another fun fact is that V2 may form complex representations like combining orientation and depth. This means that these cells in V2 respond to illusory contours, which is the Gestalt stuff from a few chapters ago. TLDR. V2 begins the extrastriate cortex, and it receives inputs from V1. Functional pathways in the visual cortex. The ventral pathway starts with midget and bistratified retinal ganglion cells and continues through the visual cortex into the inferotemporal cortex in the temporal lobe. This is often called the what pathway because it codes for object identification and color vision. So the ventral system is often called the what system. The dorsal pathway starts with parsal retinal ganglion cells and continues through the visual cortex to the parietal lobe. And it is often called the where pathway because it codes for the locations of objects and their movement. So, since the dorsal pathway codes for place and movement, it is the where system. In short, the ventral pathway flows through V2 and eventually into the temporal lobe and is concerned with object identification as well as color perception. On the flip side, the dorsal pathway flows through V2 to area MT and then to the parietal lobe. It is concerned with where objects are in visual space as well as motion. Pathway locations and functions. The what pathway? Ventral. Located in the inferotemporal cortex, which can be found in the temporal lobe. The function? Complex object and face recognition. The where pathway? Dorsal. Located in the parietal lobe. Function? MT-V5. MT is a motion detection area that is important for awareness of movement. So the where pathway is just Awareness of movement, helping you find where things are. Ooh, and a not so fun fact is that object agnosia is an acquired deficit in identifying and recognizing objects even though vision remains intact. You have a real hard time identifying and recognizing objects even though your vision is still there. That's object agnosia. And a quick note here is that the inferotemporal cortex is the region in the temporal lobe that receives input from the ventral visual pathway. One of its functions is object identification. Okay, this was some stuff that was covered in class. Book mentions it a little bit, so let's talk about it here. Functional pathways. Lesioning studies. Lesioning studies basically destroy part of the brain while keeping the animal alive. And depending on where the lesion is, this destruction may interfere with certain kinds of tasks. From this destruction, we learn what certain parts of the brain do. And this is the work with the monkey. When the lesion is in the parietal lobe, the monkey cannot associate an object as a marker indicating where food is. So the landmark task, which the monkey in this case failed, is find food in a bin closer to the landmark. But when the lesion is in the inferotemporal cortex, the monkey cannot discriminate between objects. So, object task, which is what this monkey failed at, is find food in a bin under the square object. So, TLDR, monkeys learned one of two tasks, a landmark task and an object task. Lesions to the parietal lobe only interfered with the landmark task, and lesions to the inferotemporal lobe only interfered with the object task. And this just shows that the dorsal and ventral streams are separate. So, moral of the story, the dorsal and the ventral streams are separate. Oh, and a quick note here is that any monkey that had difficulty with the landmark task could do the object task, and any monkey that had difficulty with the object task could do the landmark task. Okay. Human happenings. Alright, functional pathways. We have obtained evidence of functional pathways from humans with different patterns of brain damage. Right, here we're talking about dissociation. Dissociation is just impair one aspect of performance while leaving other things intact. People with damage to the temporal lobe have object agnosia. This is what we mentioned earlier. This is the inability to identify objects. A textbook case of this is patient DF. These people, like DF, can identify location, but if you test them on objects, they will fail. There are case studies on these. Alright, sometimes the impairments are general, which means they apply to all objects, but other times the impairments are specific, like specific to faces, difficulty identifying faces. Blind sight is another example of dissociation. Right? Blind sight is the presence of visual abilities, even though a person experiences blindness because of damage to V1. And now, when it comes to actually trying to do an experiment with this, you can create dissociation by using methods like masking. Now, we didn't go too far into this, so this is more like a fun fact here. A step up from this is double dissociation. Alright, double dissociation is more complex. To have a double dissociation, you need two different people. Alright, you find two people with two different patterns of brain damage. For example, one person with a damaged temporal lobe and one person with a damaged parietal lobe. Once you find these people, you test them, and you will get opposite results, like what we saw with the monkeys. Succeed at one test, fail at another, and then reverse for the other one. Now, this is useful because we gain more confidence in identifying the function of a particular brain region. And now, the big question. Where in the brain do all of these pathways come together to give us our common perception of the world? Oh, we don't know. Okay, that is going to do it for Chapter 4 of Sensation and Perception. Thank you all for watching. I'll see you next time. Class dismissed.